course. For those of you that don't know me, I'm Maria Abreu. I'm a gastroenterologist, and in this context, I serve as Vice Chair for Research in the Department of Medicine. And this is the day that makes me very proud because it's an opportunity for us to come together around the theme of research. And as I mentioned a second ago, hopefully inspire our residents and trainees to imagine that they can also do research and grow up to be someone who's a grand round speaker on Research Day. I want to tell you that this is the 8th year of the Gene Safety Research Day. It's named for Gene Safety because he, through generous donations, have supported this Research Day. Gene passed away actually from COVID, and the reason I think that's notable is because I'm quite sure that he got that from a patient because he would never shirk his responsibilities as a clinician. You know, he's someone that it would be hard to live up to, in that he was always available to his patients no matter what day or time. And so this is our 8th year doing that as a result of Gene and his and the generosity of him and his family. So, and apropos to today's talk, he was a cardiologist, Beth, even though he did mostly primary care. So I want to tell you a little bit about our speaker today, Elizabeth McNally, who is joining us virtually from Northwestern. And Dr. McNally knew early on that she wanted to combine science and medicine. After she finished at Barnard in New York, she was accepted into an MD PhD program at Einstein, where she was already highly published by the time she came to do her internship at Brigham and Women's Hospital, where I had to, first met her and first thought, oh my gosh, how does someone get to be so smart? So Beth was a remarkable intern resident, so laser focused and intelligent and of course extraordinarily hardworking. And Beth ultimately short-tracked out of her residency at the Brigham into the fellowship at the Brigham in cardiology, although there's so much cardiology at the Brigham that we had really all short-tracked into a cardiology fellowship at the Brigham, even as interns and residents. And then ultimately was at the University of Chicago with Dr. Weiss and is now at Northwestern. If you are familiar with Chicago, which is a beautiful city, Northwestern has the best real estate. And Beth's office is no different than that, with a spectacular view of Lake Michigan. Beth is a cardiologist, but she really, in a way, has begun this whole field of cardiac genetics and understanding the genetic and genomic underpinnings of the cardiomyopathies that we probably are seeing, and before never realized, oh, this might be a genetic syndrome that this patient has. In recognition of her contributions, she has many different grants, actually a lot of different types of grants, and not just the typical R01s, and it reflects both her commitment to training. She has a T32, but also R01s, and is part of a U01 to study cardiac disorders. She's very involved in the Muscular Dystrophy Association, because again, that also of course has cardiac manifestations and is the kind of the emblematic genetic disease that causes heart disease. But again, as a recognition of the pinnacle at which she is in her field, not only was she elected to be in the American Society of Clinical Investigation, which is the Young Turks, and then the Association of American Physicians, which are the Old Turks. In fact, she's been president of both societies and is the current president of the AAP, the Association of American Physicians. And then sort of the icing, the most recent icing on that incredible cake is that she is now the editor-in-chief of the Journal of Clinical Investigation. And that means the editorship of the journal is at Northwestern and at the University of Chicago. To me, JCI continues to be that journal that you have a monosynaptic reflex is the one that is involved in publishing truly translational work. Beth wrote a really terrific editorial about where she wants to take the journal. And I think it actually encapsulates a lot of what we should aspire to in academic medicine, making sure that we're helping reflect diversity, because if there are people that can't have a seat at the table, we're doing something wrong. And I love the notion that she's going to put a lot of emphasis on making sure that the research is published quickly and that there continues to be an emphasis on human research. So without any further ado, Beth, thank you so much for making time out of your extraordinarily busy day to have time with us, and hopefully we'll have time at the end for, you know, our faculty and fellows to ask questions. Thank you.
Thank you, Maria, for that wonderful introduction. And, you know, it was such a delight so many years ago to be an intern with you and watch your career grow and flourish, and the amazing things that you have done. So today we're going to talk about genetics and ancestry in medicine. And I'm going to give you the perspective of cardiac genetics. The reality is if I were a cancer geneticist, I could give almost this exact same talk, talking about some of these same issues in cancer genetics. We could do it in neurogenetics. We're starting to see ophthalmology genetics and renal genetics. And so what we're really seeing is an explosion of the utility of a lot of genetic testing within disciplines in medicine. And for those of you who are in training, this is a fantastic time to be learning about this, because it will be part of what you do as you take your careers forward in medicine. It also represents, for me, a terrific opportunity as a physician scientist, because I'm of the mindset all of this information that we learn in genetics, an individual patient with an interesting new mutation can teach you so much about mechanism. And as we go forward right now, especially I work a lot in neuromuscular diseases, we're seeing genetic therapies now become reality. And so that's the future, and it's exciting to be able to watch that happening. So let me do this. So just some of my disclosures. I do talk a lot with a lot of companies these days, because there is a strong interest in the rare diseases and using some of these new genetic tools to develop therapies for these patients. That's not related to any of the content of this talk today. And so we'll just start by broadly putting human genetic variation in perspective. When we look at individual genomes, we focus a lot on single nucleotide changes that are present within a genome. And so that's a single base pair that's changed from one thing to something else. And there's an enormous amount of single nucleotide changes within a single given genome. What we find is that if I sequence my genome, I have about 5 million differences in my genome compared to what the reference sequence is. And if you come from a background with more diverse genetic ancestry, that number is higher, as I'll show you shortly. If you were to take all the variation in the genome and say, well, how much is this high frequency common variation, the kind where if I look across a group of 100 people, I might find 30% of the people having a G in that position and 70% of the people having a T in that position. That kind of high frequency variation represents about 15% of the total variation in the genome. The vast majority of single nucleotide changes within each of our genomes are actually rare variants. And so that's really what makes us up. But this high frequency common variation is what we've used a lot over 20 years now in genome-wide association studies, or GWAS. These are considered to be ancestrally older evolutionary variants, and that's why they're a little more fixed in populations or within populations. And so we can use that to determine genetic ancestry. But by nature, most of these variants, because they are common in the population, they really don't have vast clinical effect at an individual level. What they do have, as you start to sum them together and take collections of these common variants, is you can get to polygenic risk scores, which we'll talk about shortly. But these stand in contrast to rare variation. So as I said, about 85% of your single nucleotide changes in your genome are actually rare, meaning they're found less than 1% of the time in the population. Some of these rare variants are the things that make up the Mendelian diseases and are considered pathogenic. And so when we find such things like a pathogenic variant responsible for a disease, we check it within the family. We use that to really identify family member risk, but it's very important for a lot of these types of variants to recognize that they are mostly associated with variable expressivity and penetrance. And so often they're associated with carrying risk. And often that risk is age dependent, depending on the disorder. And mostly these are considered evolutionarily younger, particularly the deleterious ones, because they wouldn't really survive in the population that long, especially if it affects reproduction in any ways. But this rare variation is what we use when we do genetic testing. And so in the cardiovascular diseases, we actually find genetic testing now useful for a lot of our disorders, very useful in the heart failure space for the cardiomyopathies. It's highly, so we have genes that really predict arrhythmia outcomes, and that's where we see that as being so important. We see a huge overlap of the cardiomyopathy genes and the arrhythmia genes, and I'll talk about that shortly. Gene panel testing, where we're doing a bunch of genes sequenced at the same time, focusing primarily on the coding region. We've been doing that for neuromuscular diseases now for, again, almost 2 decades. It's how I got interested in genetics, because I was long the person who takes care of the cardiovascular complications with neuromuscular disease, and the neurologist would send me patients, and the first thing I would say is I need to know what the genetic diagnosis is, because I don't know what the cardiac risks are unless I know that diagnosis. So that's really a mainstay. We really provide that information for almost all our patients. It's very rare that we have patients that we don't know what their genetic disease happens to be. If you don't find an answer by sequencing 100 genes or 200 genes, which is now easy to do through genetic testing, we sometimes go on to do more broad-based sequencing, where we look at the entire exome, all the exons, the genome, or in combination with RNA-seq sometimes of the affected tissue. And this type of approach is particularly useful for younger onset diseases. So our pediatric colleagues, for example, will very commonly do what's called a trio exome, where you do both parents and the offspring, and with the idea that that's often some recessive disease that you can pick up that way. And again, these are clinically available tests, relatively easy to order. The panel testing has come down greatly in prices, making it very easy to get, and insurance covers it. The exomes, for example, still remain relatively expensive to do. Now, most of what we do when we do genetic testing is we start with individuals who carry a diagnosis, or in genetic land, we like to call that the phenotype. And so one of the prototypes where we think about this is in the disease called hypertrophic cardiomyopathy, where a patient comes to us and they have thickening of the ventricle, marked thickening of the septum, outflow tract obstruction, and arrhythmias that may accompany that. And so we'll take that person who has disease and we will want to diagnose them genetically. So we start from somebody with a phenotype, and then we go forward and we say, well, what's the genotype that goes with it? It helps predict those people who are at different risks and different progression of diseases. I'll show you shortly. But increasingly, we have large databases of genetic information sitting side by side with electronic health record information. We've done this through the eMERGE consortium, which I'll tell you about in one moment, but we're also seeing increasing databases coming forward right now in the form of the All of Us, which is a nationwide collection aiming to get 1 million participants in it from diverse populations. And the first genome release of those All of Us participants is coming available very soon. We also have some programs called TopMed and the Million Veteran Program. All of these will enable this type of question, which is to start from the DNA information and then to say, what's the range of phenotype we see associated with that DNA? And the reason why these are different, starting from the phenotype or starting from the genotype, is because, again, of that variable expressivity of what do we actually see when we look at this specific genetic variant in the context of all their other genetic variants. We get a range of outcomes. And of course, there's social determinants of health, which changes the way these things manifest as well. So one of the sort of really original studies of combining genomics with electronic medical records is called eMERGE. It has been around. It is now in its 4th funding interval. It's sponsored by NHGRI, which is part of NIH. And it has historically had at each 5-year interval a collection of about 10 to 12 different sites. Northwestern has been privileged to be part of the eMERGE consortium since its inception. And so we are now part of this 4th, what we call E4. It's the 4th, 5th year funding interval of this. And these are the current partners who are engaged with the this version of the eMERGE network. So during its many years, the first phase, so the first 5 years of the eMERGE network really focused on genome-wide association studies in combination with electronic health information. So they were doing SNP genotyping and looking for electronic health information. And typically the cohorts have been about 25,000 people, collected about 2,000 or so from each of the various sites that are participating. The second phase really focused on pharmacogenomics and how that can impact healthcare. And then phase 3, which was when I joined Northwestern, and we've completed this phase now, was looking at 109 genes, deeply sequencing those 109 genes. And that included, which at the time was considered the 59 medically actionable genes. So the American College of Medical Genetics and Genomics identified 59 medically actionable genes. So if you have a gene mutation, it's going to change your healthcare in a variety of ways. And part of what we do in this eMERGE network is we provide that genomic information, we put it into the medical record, and we look at what the outcomes are and how does having genetic information really change the way things go forward. So I'll just tell you, I'll talk about it a little bit towards the end. Phase 4 is on polygenic risk scores, and we're getting started in that part of it right now. So the medically actionable genes, because this was phase 3, was really to talk about what happens when you sequence all of these genes and return results to participants. In general, it's about 2 to 3% of individuals happen to have mutations in these genes. And these medically actionable genes, if you look across the broad categories here, they're about half cardiac genes and half of them are cancer genes. And when we look at the cardiac genes, a lot of them are genes that we know are important for cardiomyopathies, and then some for arrhythmia phenotypes. And then a series of them are also involved in aortic disorders as well. So sort of the same spectrum that we see in our CardioGenetics clinic. And I just, when we do this analysis, when we test genetically, we have a variety of answers that we get that return to us on the reports. The most useful information comes when the genetic variant is found to be pathogenic or likely pathogenic. That means you can use that information in determining healthcare practices for that individual and risk mitigation practices for that individual who carries those variants. And then on the other end of the spectrum, we have benign variants. And most of these are determined to be benign because they're high frequency in the population. And then in the middle, we have variants of uncertain significance, which means these are rare variants, but we simply just don't have enough information to know whether they're pathological or whether they're benign. And so the variant of uncertain significance is a difficult diagnosis, particularly for the clinicians, because we like answers. I personally find the patients don't struggle with it as much as the providers struggle with it. But coming back to what actually makes these benign, benign is really determined by what is the frequency of that variant. And part of the issue is those variants have to do with what is the genetic architecture of that person's ancestry that you're seeing. So people from different ancestry backgrounds may come from very different genetic architectures, and knowing whether something is present at a high frequency in that background may not be possible. And it also equally has to do with how well that ancestry group is represented in our genetic databases. And we know that we don't have very representative genetic databases at this time. It has improved in the last few years, but we still have quite a long way to go. So this is just an example of a result that we did turn that was out of the eMERGE 3 consortium. This was a 58-year-old male who has an MYH7, which is a cardiomyopathy gene, and he has a single change which shifts an arginine at position 870 to a cysteine. MYH7 is well known to be associated with hypertrophic cardiomyopathy, and this individual did not carry a diagnosis of hypertrophic cardiomyopathy. What he did is he presented 2 years earlier with a stroke, left facial droop, and in the setting of having new onset atrial flutter. Now, the good news is he did very well. He received thrombolytics and completely recovered, didn't have any neurological sequelae. And then he got an ablation for his aflutter, and so that has done pretty well. But what he was carrying was a mutation for hypertrophic cardiomyopathy. So how well do we agree that this is in fact a mutation that causes hypertrophic cardiomyopathy? Well, this is part of why I selected this as an example, is this is a variant we know, a gene we know well, and variant we know well. It has undergone what's called expert curation. So an expert panel has reviewed all the data on it and agreed that this is a likely pathogenic variant. So there's no doubt that this is in fact the interpretation is correct. And again, we know very well these are linked to hypertrophic cardiomyopathy, and yet this individual had no history of hypertrophic cardiomyopathy. So what we also do is we use a large database called NOMAD, which is run by the Broad Institute, and it is an aggregation of any large data sets of exome or genomes that can come together. And you can query this freely. The new version is going to be coming out soon, and it's going to have, I just heard a talk on it the other day, it's going to have a lot of really cool new tools and variants that you can use. But this is what we look at when we look at myosin heavy chain. The gene is actually going from your right to your left. And these are all the different exons of the gene being sequenced. And what they have now is a bar that shows you where variants are positioned. And what I'm showing you here is individuals that are pathogenic and likely pathogenic. And so more of the variants that are pathogenic or likely pathogenic fall in this first half of the gene that again is on the right-hand side of your screen. But where all these red bars are, you see there's a cluster of this area of this encodes myosin, which is the motor protein, very important for contraction of the heart. And this falls right in what's called the converter domain of myosin. So it's critically important for how myosin uses ATP to be able to contract the heart properly and with proper energetics. And this 8-7 variant falls right in this converter domain. And there's been some work that says variants within this region have an unbelievably statistically high chance of being pathogenic. And so again, further support that this is a pathogenic mutation. If we look at this person's echo, he did have some septal thickening, and you can look at this immediately and say, well, there's a little bit of thickness all the way around that heart and concentric left ventricular hypertrophy. Sometimes we see that in people who have a history of hypertension, but sometimes we see this as a mild form of hypertrophic cardiomyopathy. And on MRI, his septal thickness was actually read as 1.4 centimeters, and he had some delayed enhancement as well as enlarged atria, which is fairly put like a mild version of hypertrophic cardiomyopathy, yet his major presentation was really one of atrial fibrillation. Now, going back in his history, his father had died suddenly at age 62 of a heart attack, and his father's mother died at age 60 also suddenly, which portends that maybe there's some even greater risk associated with this variant for some more serious arrhythmias like ventricular arrhythmias. And so I wanted to draw your attention to this, which is called the SHARE Registry, Sarcomeric Human Cardiomyopathy Registry. A great group of investigators got together and looked at about 4,500 hypertrophic patients, 2,700 of which had genotype information. And the real take-home message of this is that if you see an individual with hypertrophic cardiomyopathy, about half of them are going to turn out to have sarcomere gene mutations. Myosin's part of the sarcomere. And their risk and their trajectory of disease is different if you are sarcomere gene positive. And this is data from the SHARE registry showing that their heart failure composite is shown here. So again, individuals who carry sarcomere mutations are at greater risk for developing heart failure. Interestingly, they're at much greater risk of developing atrial fibrillation and also of developing ventricular arrhythmias. And I know this is what many people worry about in hypertrophic cardiomyopathy, but frankly, the atrial fibrillation risk is much, much greater than the ventricular arrhythmia risk. And so that really underscores that perhaps one of the presenting diagnoses with hypertrophic cardiomyopathy is in fact atrial fibrillation. And it has caused the field to really think a lot more about things like atrial myopathy and how do these mutations affect the atria, or is this all secondary from the ventricle? But it's led to a whole different way of thinking about it. So more recently, this paper was published from TopMed, which is again a large medical genomics sequencing consortium, looking at individuals with early onset atrial fibrillation. Early onset atrial fibrillation is defined as being AFib that occurs before the age of 66. So it's not terribly early onset, it's just younger than age 66. And I'll point out that the mean age of developing atrial fibrillation in males is actually 67. So we're talking about half the males that have early onset AFib, and that's across the broad population. What this group found, looking at a cohort of about 1,200 early onset atrial fibrillation patients, was about 10 to 11% of them turned out to have mutations in cardiomyopathy and arrhythmia genes. And dominantly, this was really in cardiomyopathy genes, with titin and myosin heavy chain 6 being the biggest contributors to this risk. So it's really suggesting that the first presentation of AFib has about a 10% chance of really being linked to cardiomyopathy, and that those individuals are at risk for developing cardiomyopathy over their lifetime. So some of the limitations of these large data sets at this point in time, whether it's TopMed, whether it's the UK Biobank, or some of the larger data sets that we've looked at, is that they are extremely skewed towards individuals of European ancestry. And so we don't always have that information about what it looks like in non-European ancestry individuals. And so I point out this second paper here, which looked at a cohort of non-European ancestry individuals, a mix of individuals who identified as being Hispanic as well as being African American, turned out to have a similar range of early onset atrial fibrillation in terms of age and association of rare genetic variants, again, primarily in genes like titin and myosin genes, and demonstrating that this is true not just in a European ancestry population, but really in non-European ancestry populations as well. And so if we break the world up again into these higher frequency variants and these lower frequency variants, we can get a lot of great signals to think about pathways that are important for the development of heart failure. So among these common variations, we pick up genes like this, which the SINPO2 and myosin is really linked to AFib, also in the setting of failure. BAG3 is a really interesting gene, and a whole series of others. The rare variation genes that are implicated in the genesis of heart failure include largely some different genes. MYH7 we talked about, MYBPC3 we'll talk about in a second, but there are some genes that show up on both lists, like BAG3, for example. And so that's really becoming signals for people to be thinking about new targets for the treatment of heart failure. So I mentioned that we have a lot of genes that can cause cardiomyopathies. We have many different genes that can cause arrhythmias, and that has led to the practice really of looking at cardiorhythm gene panels. This is our practice in our CardioGenetics clinic, which is we tend to order the broadest panel we can get. The price is the same, and so you might as well get all the information you can. You do increase the risk of getting variants of uncertain significance, but you also maximize the chances of actually getting a result for your patients. So we talked about myosin heavy chain, and in the next couple of minutes, I wanted to talk about myosin binding protein C3. So myosin binding protein C3 is another gene that's well known to cause hypertrophic cardiomyopathy. What's best described is premature truncations in this gene, and this is most typically in the setting of having dominant inheritance. So one copy of the gene that can be inherited either from the mother or the father that carries a premature truncation and an increased risk for developing hypertrophic cardiomyopathy. There are sometimes individuals that have two hits in their myosin binding protein C3 gene, so one maternally inherited, one paternally, and those usually have very early onset, childhood onset disease, and quite severe. But what I wanted to focus on was a particularly really interesting intronic 25 base pair deletion. So this 25 base pair deletion sits right towards the end of the gene, so right after exon 32, but before the end of the gene. And what the presence of that deletion does is it disrupts splicing in a variety of different ways, so that individuals who carry this have a partial knockdown of one of their copies of the gene. So it's what we refer to as a risk allele. It doesn't have the highest risk as having a hypertrophic gene, but it's more like an intermediate risk allele gene. A lot of what we see in the cancer genetics was risk alleles, and so this is exactly like that. What's really quite interesting about this is it is heavily distributed. This delta 25 allele is very heavily distributed in South Asia, and also in East Asia, but then has sort of made its way around the world as we've seen populations migrate to different parts of the world. But it was first described in South Asia quite some time ago. In 2009 was the first description of it. Let me just show you here. This is some of the odds ratios associated with that first discovery in 2009. So an odds ratio of 7, which is an incredibly high chance of developing heart failure. So again, at least in these first populations where it was looked at, it was really a risk allele. Where it's been looked at, and it's been looked at in a number of South Asian and East Asian populations, it sits at about 4 to 5% of the population carries this variant. So again, identifies individuals at risk of developing heart failure. We did a study a few years back with Sakthi Sadiapan, who at the time was at Loyola. He's now moved to Cincinnati. And Sakti's been very interested in South Asians living here in the United States. Some of you may know that the South Asian population is one of the fastest growing populations we have here in the United States. And so Sakti had collected 2,400 South Asians living in the US, and we did genotyping, and we found that those individuals, about 6% of them carried the Delta 25 risk allele. We did see the occasional individuals who had 2 copies of the gene, so they do not have the severe early onset phenotype. They just, it does exist in the normal distribution you would expect. And then he did echoes on a subset of these Delta 25 carriers to say, well, what do these patients look like, these participants? Do they look like they have features of hypertrophic cardiomyopathy or mild hypertrophic cardiomyopathy? So, and the short answer was, it wasn't terrifically impressive. So here is E to E prime, which is a diastolic measure, and you see it really isn't different between carriers and the non-carriers. The fractional shortening was a little bit ticked up in the Delta 25 gene carriers, and this represents like a very early phase of hypertrophic cardiomyopathy. Not everything that's going to go on to become pathogenic, but this hyperdynamic state where the heart's actually beating a little bit more and has an increase in its function is again reflecting that energy inefficiency that's present in these hearts, and is much more clearly seen in diagnoses of real hypertrophic cardiomyopathy, but a little bit of hint of signal, but again, not terribly impressive. And the ejection fractions were not different. And so Megan Pucklewartz, who led this study, decided to do a bunch of sequencing on some of these non-carriers and carriers and say, well, is there other additional hits that could explain anything? Did anything else happen in these genomes? And so what she found was a second hit in myosin binding protein C3 called D389B. So this had, at the time, had not been previously described. It was not present in any database at the time, largely because South Asians were not well represented in the database. And what Megan found was that the D389B sits here in exon 12, and it sits on the same copy of the gene as Delta 25. So while there are nearly 100 million people worldwide who carry this Delta 25, only about 10% of them have this D389B. So it arose on the background of the Delta 25, and again, fixed at about somewhere around 10% of the population. The D389B, from a protein standpoint, sits in a really important part of the molecule that interacts directly with myosin, so the motor protein of the heart. Again, we found it in about 10% of the carriers were carrying the Delta 25 plus the D389B. And the very simple way that you know that they're on the same copy of the gene is you just do some family studies, which shows that here's a mother and she's passed both of these on to her children, which means it has to be on the same copy of the gene because the other copy of the gene came from dad, who was a non-carrier. But when we looked at the echo data on people who had D389 plus the Delta 25, these individuals had increased ejection fraction and increased fractional shortening much more clearly. So it was describing that the signal of this sort of early hypertrophic state, or perhaps more associated with the risk of heart failure, was really coming from having the 2 hits together. So it really does begin to underscore that there's contribution of more than one gene. And this was just some molecular modeling that shows that D389 sits on the outside of the molecule, where again, it's positioned to interact with myosin itself. We generated some induced pluripotent stem cells from these participants and made them into cardiomyocytes. And the cells were larger in size, similar to what you would see in hypertrophic cardiomyopathy. They had a lot more irregular calcium transients, which can be construed as perhaps explaining why they might have some increased risk for arrhythmias. So can genetic information begin to predict who's at risk for heart failure? And I would suggest yes. I think we know a lot about rare variation, that it does a very good job of identifying individuals who are at what we call stage A. They are high risk, they don't have any symptoms, they may not have any evidence of structural heart disease. And certainly in our clinic, some of the things that we offer to these at-risk individuals is tighter management of their blood pressure, and we're much more meticulous about their cholesterol management, but we try to take preventive steps with these patients to see if we can change their outcomes relative to their older relatives who developed heart failure. So in the next little part, I want to talk about diversity in our biobanks, because as I alluded to before, it's one of those areas that we need to do better in. And so this was a study that was an offshoot of our eMERGE consortium work. We have our New Gene Biobank, which has about 15,000 people in it. We have reasonably diverse representation in our biobank, and we had the opportunity to get about 900 whole genome sequences done. But what we wanted to do was to select a group of individuals that were diverse and sort of equally represented in this cohort of 900 whole genomes. So about a quarter, so we selected genomes to be about a quarter of African ancestry, about a quarter European ancestry, about a quarter Hispanic, and then about a quarter of individuals who just identify as being of more than one background. And we do this by asking what the grandparents are, as opposed to just say, how do you self-identify? It was done by genetic counselors and documenting what the self-identified race was of their grandparents. And so that's how this was determined. The average age of participation differed a little bit for the groups, but not hugely. And so a critical part of this is to recognize that the amount of variation in a person's genome does vary with what their ancestry is. And here I'm using Ancestry to talk exclusively about their genetic information. So as I said before, people of European ancestry, and I'm going to show you here how we look at this now using principal components analysis, but this little blue dot is the 200 people in our cohort who were of European ancestry. They largely pile up on top of each other, which means they are more interrelated to each other than people who identify of Hispanic ancestry, shown here in green, or people who identify of African ancestry, who are shown here in the orange color. And then this purple or magenta type color represents people who are of mixed ancestry. And we refer to these as the PC curves of ancestry. So you can see that they really are much more diverse, not only from the people who are of European ancestry, but within these groups, it is much more heterogeneous. And that's graphically displayed here on the left. For the individuals, what that means that if you're of European ancestry, you have less than 5 million variants in your genome that differ from the reference sequence. But if you are of African ancestry, you have close to 6 million variants in your genome. So a lot more variants are present in these genomes. Similarly, Hispanic ancestry and of course mixed ancestry, you see those same trends. So that means the genetic architecture of these groups is different and has to be taken into account. One of the earliest diverse populations that got looked at from a genome-wide level was looking at the 1,000 Genomes database, individuals collected from around the world, so not necessarily African-American, but African. And so what we did was we backfilled that data and aligned our Chicago-based biobank against the 1,000 genomes. And you see that we actually compare pretty well. So we see in Chicago, I'm sure much like you in Miami, here in America, we see the same diversity in our populations as if we were taking care of people from all around the world. And so that's just something to bear in mind, that our populations, our genetic ancestry within our populations we care for here in America is really very, very diverse. And so looking at pathogenic variation, that was one of the questions we wanted to ask across this cohort of 900 genomes. We did in fact find individuals who carried mutations associated with long QT syndrome. When we looked in their electronic health records, that evidence of long QT syndrome, so long EKGs, across a broad age range, across a range of ethnicities, and none of these individuals carried a diagnosis of long QT, so we were underdiagnosing these individuals. Similarly, we saw a group of individuals who also had mutations consistent with hypertrophic cardiomyopathy. Only a few of them had echoes, and at least 2 of them had some increased wall thickness, again, suggesting there may be some underdiagnosis going on there as well. So what I talked to you about was that individuals who come from African ancestry backgrounds have increased variation. Well, what about variants of uncertain significance? So this is saying when we break down that variation, do they have more variants of uncertain significance? And the fact is, when we look across all of their genes, people of African ancestry have increased variants of uncertain significance. And notably, they have a lot more of these variants of uncertain significance in these medically actionable genes and also in the cardiac genes, which means it becomes harder to interpret some of our genetic testing results in individuals of non-European ancestries. Similarly, at the time we did this study, we said, well, what about variants that aren't even reported in any databases, representing the idea that we don't have deep enough representation in our databases. And similarly, just those unreported variants were also increased in some of these different backgrounds in all of the genes. The good news was it wasn't so bad in the medically actionable cardiac genes, indicating we were doing a little bit better in representing there. But what Tess Pottinger was a student who led this study, and what she wanted to do was begin to ask that question, well, those variants of uncertain significance, I told you those were increased. Does that correlate with anything? And so she came up with a fairly clever way of looking at this, which is to look at the trajectory of changes in their echocardiogram data over as much as 14 years. That's as much as we had in the database. And what she found was individuals who had variants of uncertain significance had a change in their dimensions in a different pattern than if you didn't have any of these variants of uncertain significance, which means that, again, some mild signals of progressing towards heart failure if you have variants of uncertain significance. And why that's happening is because some of these variants probably are pathogenic. And that's what she was really picking up with this. And so when she began to look at what was really some of those variants that were driving this signal, it turned out to be our old friend myosin binding protein C3, which is shown here. And that was the gene that had the majority of the variants in it. So just a couple little last things. So in this next phase of eMERGE, we'll be doing polygenic risk scores for common diseases. We know that these work better, similar to what I just told you, same story. These work better when they're done in individuals of European ancestry, but a major initiative that we have here is to try to apply these in diverse populations and to try to build some of those data sets so that our prediction of these common diseases gets better in groups from diverse populations. These are just our participants at this point. We've spent a lot of time going through this workflow, and we're just getting ready now to broadly start the recruiting as it begins in the next few weeks. And so just a couple words about ancestral history. Part of why we have these different genetic architectures across different groups is because of human evolution. So humans came out of Africa, the brief stop off in Europe with some mixing with Neanderthals, moving into Central Asia, down into Australia, and then of course, ultimately into the New World where there has been mixture from European and African genomes. And that's part of why we see this difference in ancestry as we look across different groups. And again, groups that we see here very well represented in the United States. And so there's been a push, certainly with respect to ancestry, talking again about the genetics, to stop using self-identified race and to use race to mean the social construct and to use ancestry to mean the genetic variants. And where you are looking at a biological trait and talk about what you think is a genetic contribution, to use the term ancestry when you are referring to that. And there was a nice piece that was written last year in the New England Journal of Medicine, really pushing this point, which is starting to separate the concepts of race from genetic ancestry. And so following on that, genetic ancestry, as we'd like to be using it now, is to really refer to using genetic determinants to assess relatedness among populations. And this is, again, showing you this principal components analysis where European ancestry groups are more tightly related. There's now conventions that you should use at least the first principal component or the second principal component or even the first four principal components of ancestry to really be when you're referring to that in the populations you're studying. Now, some of the tricky parts of doing this is that the genetic variants for determining ancestry have shifted over the years, and that's with the availability of more and more data. So in the early days, people tried to do this looking at mitochondrial DNA, little tiny genome, easy to sequence, and that got you at a lot of maternal information because mitochondria are maternally inherited. It's been done with the Y chromosome, it's been done with copy number variants, but the convention right now is to use single nucleotide polymorphisms and to use them as they really do pepper across the entire genome to be able to grab that. But remember, the number of SNPs that you study and their distribution across the genome will influence the results when you try to do an analysis like this. And part of that is because ancestry isn't exactly uniform across the genome. There are bits and parts of the genome that may come from different ancestries because, again, we're all a big mixture. And so we're starting to hear a lot of good conversations about local ancestry when talking about particular gene regions and what that may look at. But the key take-home point is that ancestry is not race. And so what can happen by not having adequate representation in our databases is that we can have genetic overdiagnosis. This was a paper from 2016, which really highlighted for hypertrophic cardiomyopathy that there had been a group of individuals who were of African ancestry who had been told they had genetic mutations causing hypertrophic cardiomyopathy. And in point of fact, when we had better databases with better representation of African ancestry, those turned out to be at too high a frequency within African ancestry populations and really was overdiagnosing these individuals and telling them that they had pathogenic variants that they did not have. Similarly, we've seen examples of genetic underdiagnosis. And the main way that we see that is that when we do our gene panels, we are more likely to get variants of uncertain significance in particularly African ancestry, but sometimes Asian ancestries as well. And we are a little more likely to see inconclusive results in some of those results as well. And I will say this was from a couple of years back, and I feel like I personally have seen this improve over the last few years where we're really starting to get better databases that help us represent better. And so it's improving our ability to interpret. And so I feel like we're closing the gap on this one a lot more. And polygenic risk scores, as I mentioned before, this is using, you might use 100 or 200 or 300 SNPs across the genome. These signals were derived from genome-wide association studies, and then they're summed together to predict risk. So it uses an array-based strategy to do it, and it is similar to ancestry testing. And I like to highlight this paper because this is a really interesting example where they looked at blood pressure control and how well it did or did not correlate with having genetic West African ancestry. And the take-home point was that blood pressure and blood pressure response to medications really didn't correlate with how much West African ancestry was present, suggesting it was really the social determinants of health that were much more important in determining how well blood pressure was controlled and response to medications. And so I think it's very important when you're looking at even something as simple as blood pressure to acknowledge that it's got a contribution of some genetics, but it's got a huge contribution of the social determinants of health. And as we go forward, we really do need to think about how to break these different components down. And so I was privileged to write a little comment on this with Dr. Yancey, who's our chief of cardiology here, and other additional commentaries, which is to say that as we take care of, as we look at population, we need to use genetic ancestry. We need to break down social determinants of health in that same way so that we can better inform our individual patients what are the parts of their lifestyle that they can change, or how can we better manage them to control their blood pressure a little bit tighter, because maybe that's important for what is predicted by their genetics. And so that's certainly the way we approach things in our clinic. But we have, again, a lot more to do here to get better at doing this. Some of you may know this, which is when we look at genome-wide association studies, again, European ancestry individuals here are shown in blue, and you see that the vast majority of these studies, this is up until 2017, really included just individuals of European ancestry. I'm happy to say that if we took this curve out a little bit further, we are starting to see much better representation of African ancestry, Middle Eastern, Latino, and Asian ancestry, which again is a very broad group of individuals. And ultimately, we'll be able to subdivide these categories and be able to do better matching and have better information. It's one of our big pushes right now in the eMERGE consortium, because we are aiming to recruit about 40% non-European ancestry individuals. And so what we want to be able to do is give accurate information to those participants. So just one last comment about the giant gene Titin. Titin is a huge gene. It's made up of 360 exons. It's really important for heart function. And what's so interesting about Titin is it's about 1% of the population. 1% of the population carries a premature stop sign on one of the copies of the gene, placing individuals at risk for developing dilated cardiomyopathy and heart failure. And so this study, which came from the Geisinger and UPenn Biobank, it was published in 2019, did that similar analysis that I mentioned early on, which is they had a lot of sequencing done in their data, in their biobank. And so they could say, well, if we take all the individuals that have tightened truncations, what's the clinical diagnoses that we see associating with having a tightened truncation? And so it was somewhat reassuring. This was the Geisinger part of the database. Again, it's going to be pretty heavy European ancestry and Geisinger. You see cardiomyopathy coming out of this data set. You also see our friend atrial fibrillation showing up in this data set, and of course, a variety of other arrhythmias. And what was interesting was when the UPenn Biobank broke down their data set, which had more African ancestry or self-identified Black individuals in it, they did not see the signal for dilated cardiomyopathy. And so this has begun to create some ideas among some geneticists of, well, maybe tightened truncating mutations don't really cause dilated cardiomyopathy in African or Black individuals. And so I would really pause before making any kind of conclusion like that. In fact, I push back pretty hardly on those groups, because I think we also have to be really careful when we're looking at age-dependent disorders. We know you're more likely to get heart failure or be diagnosed with cardiomyopathy as you get older. And if we don't have adequate representation in our older populations of individuals who are Black or who are different backgrounds, which we know we don't have, right? If we look at groups of individuals who are over the age of 65, it becomes even more disproportionately white individuals. That may cause us to miss some diagnoses. And so that's what I think we're actually seeing here, which is that we just don't have adequate representation of older individuals who are Black. And if we did, we would see these diagnoses. And so this is our genetic reality. We are both common and rare variation. All of it is positioned to help predict what risks we carry in our lifetime. And as a cardiologist, I can tell you there's a huge amount that we can do to reduce these risks. And just in conclusion, genetic testing is incredibly useful to us to identify individuals at risk. We are still doing a lot in the context of families, but increasingly we're seeing polygenic risk scores start to come into practice as well. Genetic testing is more likely to yield uncertain variants in some ancestries due to diversity within that ancestry and inadequate representation in our biobanks, and this is starting to get better. Clinical manifestations associated with any given genotype are likely much broader than any initial report you might read, and they may vary also with ancestry and most importantly with age. I think that's probably the biggest determinant. And genetic determinants of health should really be considered in the context of ancestry, and importantly, as we are now recognizing every single day, the social determinants of health. And, you know, sort of my vision for the future is that as we take care of patients, we not only will have genetic information that helps predict outcomes, but we'll have much better metrics to really quantify social determinants of health and be able to use that information again to guide patients towards lower risk practices that keep them happier and healthier longer. And so with that, just want to point out some of the people who did all this work. This is Tess Pottinger. She was a graduate student who did a lot of the work in the biobank, and she's gone on to Columbia University where she's doing her postdoc. Megan Puckelwartz is our really lead genomicist, a fantastic faculty member that I've had the privilege of working with for a lot of years. Lorenzo Pesci is our lead genomic statistician. And then of course, the rest of my group and my collaborators from eMERGE. And so with that, I think I left a couple minutes for some questions.
Dr. McNally, that was truly elegant and wonderful and a beautiful grand rounds. Thank you so much. Maybe I can start. We have a few minutes for questions. The discovery of new genes, and we have a panel of genes that you screen for and variants in those genes, but what would happen if none of those genes showed an abnormality? How would you identify a new target, a new gene that was previously unknown?
So I think that's sort of the question a lot of geneticists get asked right now, whatever the phenotype is, which is to say, well, how much is new gene discovery that we didn't get done yet? And then the harder ones is, you know, the variants are in the genes and we don't know how to recognize them. So that's a part of what's going on. And then some of the disorders are really combinations of genes. So I think, you know, many people feel like the number of new genes is getting smaller and smaller and smaller every day. The way we go after that, the pediatric space, again, that trio exome where you're doing the two parents together with the offspring, we do that on young adults that have disorders. That's using family information and who has disease and who doesn't have disease becomes the primary way we go after that. And then the equally important part of that, when you find a variant and you're suspicious about it, which is then going back to the bench and doing the modeling of that, which we increasingly use iPS cells and build different models of these disorders.
Thank you, Dr. Abreu.
Beth, actually, I think you partially answered that question. So for these variants of unknown significance, how predictive are any of these iPSC models to try to understand whether they just, is this ancestry or is this really kind of a pathogenic variant?
So anybody who's, we've been doing a lot of IPS work for probably about 7 years now. And, the challenges of doing IPS work are exactly the challenges when we sit as doctors in the office looking at patients, which is every IPS cell is different, right? It's got all that genomic background. In addition to there's technical variability that happens with IPS cells. And so that's the really hard part. And, the convention in the field is to try to site specifically correct whatever that variant is and show you've lost whatever cellular phenotype. And I think we're realistic with ourselves that when you go in and try to do a site specific mutation in CRISPR and you've put those cells through a lot of selection, you've changed them in great big ways. And so I think there's a lot of work still to be done on the modeling. Now, the flip side of that is I also live in the world where we're watching these genetic therapies come forward for patients. And so there's also a world where we build cell models of patients with the idea that we're using that platform to test these therapies. So whether we're skipping exons or doing gene therapy or doing gene editing. And so I think we'll get better and better at doing that over time, and it will move from being a laboratory research-based test to being actually a clinical test at some point. That's how we will adjudicate some of those.
Thank you. Dr. Comanduri, can you unmute yourself and answer your question?
Yes. Hi, Dr. McNally. Beautiful talk. And I'll just tell you a little anecdote. I had probably a benign episode, but then got tested for odd reasons for Brugada syndrome and ended up having a missense mutation in the SCN5A gene, which led to me having a provocative test, which was negative. So I think the whole thing was, one of these things that happens in this era where we learn about genes, and I'm South Asian, and there are some thoughts that this may happen, in individuals of South Asian ancestry as well. And curious as to what your thoughts are about what happened to me is likely to happen to more people, right? As we understand these genes and yet fully don't have the population-based studies. What do people do, and how do we deal with the societal implications of this kind of testing that puts us in an odd situation in terms of what we know and what we don't know?
This is why the American College of Medical Genomics is so aggressive about really not wanting to be too aggressive about overinterpreting variants of uncertain significance. Truly, the majority of those are likely to be benign and not pathogenic. And so that's where, there's pretty hard push from them to not overinterpret variants of uncertain significance. SCN5A is a great example. It's an ion channel gene. And there are many people who look at things like that and say, well, that's an opportunity to develop a real cell-based or an in vitro assay where we could go through and really test every variant and know what function is. And so I think there's a lot of push right now to do things like that. And again, a lot of that work sitting in the research laboratories, but there's the capacity to convert those over to actual clinical tests. And so, hopefully the future will hold something like that, and the next person doesn't have to go through the sort of odyssey of you have a bus,
So Dr. McNally, thank you so much for being with us today, even though it was virtual. And thank you for that beautiful picture of Chicago River. We hope that we'll get to see you in person soon. Dr. Abreu, thank you for such a good choice of an invitation. I want to remind everybody, as Dr. Abreu has done in the chat, that our posters begin from 2 to 4 p.m. and then the award ceremony begins promptly at 4:30 p.m. with refreshments on the Science Quad. So looking forward to seeing everybody there. Again, thank you, Dr. McNally, and thank you all for participating thus far in today's activities. Bye, everyone.
Have a great day, all. Bye.
Thank you, Beth. Thank you.